yours.
Thank you very much. Thank you for the kind introduction. And I'd like to show you today the line of research we have started in my laboratory some time ago. It's focused on the central part of the amygdala. But I would like to ask you to interrupt me if something is not clear. I'm aware that it's maybe probably for you it's completely novel subject, so please don't hesitate to ask the questions. I'd like to start with introducing my favorite structure, which is the amygdala, and in particular its central part. So the amygdala is a small structure located deep into the brain in the medial temporal lobe, and even if it's small, it's functionally and morphologically heterogeneous. There are two main parts, the basolateral one, which is we say that it's cortex-like. It means that about 80% of cells are excitatory neurons. And another part, including the central amygdala, which is striatum-like, which means that about 80% of cells are inhibitory neurons. So they are, morphologically, these parts are different. And the amygdala as a whole, we know for a long time now that it's crucial for processing of emotions. When we remove the structure from the brain, we see a lot of emotional changes. It was done firstly in monkeys, so when the structure was removed, they started to be fearless. So they were not afraid of the objects of which they are naturally afraid, like snakes. They started to try to eat objects that are inedible, or they were also hypersexual, and their social behavior was changed. So many different kinds of emotional behaviors were affected by removing the structure. Then people started to do lesions of different parts of the amygdala, and we realized that they have different functions. And the basolateral part of the amygdala and its functions are relatively well known. It's involved in fear conditioning, but also in appetitive or food-motivated learning. But we know much less about the central part of the amygdala. So we decided to focus more on this part. So here I would like to briefly tell you what we knew before we started this project. So we know that the central amygdala is involved in modulation of motivation for food and drug rewards, and that its stimulation recruits the mesocorticolimbic circuitry. So it's considered to be a part of a reward system. But also it's important for fear conditioning. And for a long time, it was not clear why so tiny structure can control so many different behaviors. So different as, let's say, pressing the lever to get food reward, or freezing behavior, which is an expression of fear in rodents. But then the studies showed that there are different circuits of cells. There are different groups of neurons that can be distinguished by molecular markers. So there are cells, there are neurons that express, for example, corticotropin-releasing factor or somatostatin, which are not overlapping. And when we manipulate these cells, for example, optogenetically, we can see different responses. So, for example, it has been shown that optogenetic stimulation of CRF expressing neurons results in active fear responses or affects lever pressing for a reward, and manipulation with somatostatin neurons affects passive fear, that is freezing, or drinking sucrose solution. But still, when you look at the effects of this manipulation, we see very different responses that are affected. Manipulating with the same population of neurons can affect both fear and food-motivated responses. So the question is, how is it possible? And I think that the problem here is that using markers, they are just markers. They are, as far as we know, they are not used for the function of these particular groups of neurons. What is closer to the function of the neuronal populations is the connectivity of these neurons, because its inputs and outputs of the neuronal cells or groups of cells actually determine their function. So in the project I'm going to tell you about, we focus on the projections of neurons in the central amygdala that they receive from the two parts of the cortex, anterior cingulate cortex and orbitofrontal cortex, which are important for processing of rewards, and in the pathway from the central amygdala through the ventral tegmental area to the anterior cingulate cortex and orbitofrontal cortex. So this is just, we decided for one of many possibilities, because central amygdala receives many different projections and sends many different outputs, control different output structures, but we just had to decide for something. It's not possible to test everything. So I would this is kind of a disclaimer just to show you that we tested only part of this very, very complex system. Okay, and I thought that we knew that central amygdala is involved in motivation for food, but it's not, we didn't know much about its role in social motivation. And social interaction is rewarding. We know that there are, well, different types of social interactions, but when we think about positive social interactions, they are rewarding. We know that they are interacting with another individual leads to increase in dopamine release and provides positive reinforcement. In rodents, we observed that if the animals can have an opportunity to interact with others in one of the places of the cage, they create place preference. So they like the places where they can interact with others, and is one of the proofs that their social interactions are rewarding for them. They are interested in approaching other individuals and initiation and maintaining social interactions. And there are like two phases of this process that can be, well, we suspected that they can have different neuronal bases, so that's why we analyzed interactions of our animals, looking separately at the initiation, that is approach to another individual, and maintaining of social interactions. So several behaviors that serve being in a close contact with another individual. So we started this project asking two questions. First was whether the central amygdala is involved in social interactions, and if so, whether the same neuronal circuits that process food rewards are also involved in social interaction. Part of the studies were based on manipulation of cells that express CFOS. So we decided to do it because our old data showed that it was the data that I collected during my PhD project. They showed that the CFOS, which is a marker of neuronal activation, is expressed in the central amygdala when animals work for a food reward. So we trained the rats in the Skinner box. They pressed the lever for a food reward, and then when they learned the task, we introduced a noise, the sound that signal either continuous food reward, that is every press was rewarded by a food pellet, or a food shock. And what we found was that this increased value of a reward induce a lot of CFOS expression in the central amygdala. CFOS is a protein, is a part of a transcription factor, which is expressed in the nuclei of the neurons that are activated by some kind of stimulation, in this case by pressing the lever for food. And this expression was higher than in the control group and than in the group that received food shocks. So we knew that we have a group of cells that express CFOS, and we can use this in the tools we applied in our study. So I will show you in a moment what the tools are. So as I said, we firstly ask whether the central amygdala is important for social interaction. And to test it, we used familiar animals, so they were co-housed in the same cage, they knew each other. Then they were separated for 3 weeks and then reunited. And we know that such reunion causes activation of the reward system. But we didn't know whether the central amygdala is activated. So firstly, we checked activation of the structure, and we found that similarly to pressing the lever for food, we also saw the clear activation measured with the CFOS expression in the central amygdala. So then we asked the question what the cells are actually doing. And to answer this question, we used a construct, a genetic construct that is controlled by CFOS promoter. So we were able to express channel rhodopsin to optogenetically stimulate these cells, only those that express CFOS here. So the procedure was as before. They were familiar animals, were separated, then reunited, and this reunion stimulate expression of CFOS in the central amygdala, CFOS together with channel rhodopsin. So on the next day, we just reactivate the cells that were involved during this social interaction. And we saw that this reactivation resulted in an increased social contact. So at this point, we knew that indeed these cells are important for social interaction. Manipulating these cells, we can affect how the animals interact with each other. So in the next step, we decided to test these projections, or to look which populations of neurons in the central amygdala are involved in social interaction and pressing the lever for food. And this is the general scheme of the experiment. So firstly, the animals were trained to press the lever, and they were at the moment of when the real experiment started, they were well trained, so they really know what to do to get food. Then we injected the construct, the genetic construct that allow for chemogenetic manipulation of the projections. So from the ones that I mentioned, for example, from the anterior cingulate cortex or orbitofrontal cortex to the central amygdala. And then the rats were isolated and then tested, firstly in social interaction test and then in the progressive ratio test to assess their motivation to get food reward. So we look at the cells that we call social cells or food cells. And firstly, we look whether the projections that we were interested in from the anterior cingulate cortex and orbitofrontal cortex in the central amygdala were activated via the social interaction or pressing the lever for food. And we visualized the neurons that express CFOS. This is a transgenic animal that express this green fluorescent protein that you can see here under control of CFOS. So we see which neurons were activated, and the animals were injected with the fluorescently labeled transport tracers, either to anterior cingulate cortex or to orbitofrontal cortex. Then we analyzed how many neurons that are activated receive the projections. So we saw that subpopulations of cells, the social cells and food cells, indeed receive the projections from the cortex, both from the anterior cingulate and orbitofrontal cortex, though those from anterior cingulate we saw more projections from the anterior cingulate cortex. So then we ask the question, what will happen when we block these projections? And we use the construct that allowed to specifically label the projections. So part of the construct was injected either to the anterior cingulate or orbitofrontal cortex, and another part to the central amygdala. And the part that was injected to the central amygdala was retrogradely transported to the cortex, so only the neurons that got both parts of the construct had the construct that were active, and that we could manipulate only those neurons that project from the cortex to the central amygdala. Then we measured the social interactions in the animals that were received injection of C21, which is which activates this construct that is an inhibitory one. So we inhibited these projections from ACC or OFC, and compare the behavior of animals to the ones that received injections of saline. So the control animals that had in which the projections were not blocked. So we look at the initiation of social interaction, so how the animals approach others and start the interaction, and a number of behaviors that serve maintaining of social interaction, like positive reaction to the partner that is approaching, crossings, but also we look whether the animals are trying to stop the interaction. If the partner is approaching, sometimes the rats, if they don't want to interact, I will show you a movie in a moment. So if they don't want to interact, they are pushing back the partner. So we analyzed different behaviors, and this is what we saw. So blocking these projections, so the circuits that receive the projections from the ACC affected maintenance of social interaction, but not initiation of the contact, and resulted in increased blocking of the response. But when we blocked the orbitofrontal cortex central amygdala projection, we saw the more profound effect, even if the projection is more sparse anatomically. We had the decrease in initiation, maintenance, and the increase in blocking of social contact. So they were not interested in social contact whatsoever. So here there are examples of this avoiding behavior. So here, this red rat is we call it focal rat, so the one in which we blocked the projections. Okay. So as you can see, usually the rats are following their partners, but here this rat is trying to avoid the contact and it's not interested in his partner. And here is this pushing back behavior. When this green rat tries to initiate the contact, the red one, the red one, okay, try to show you again. The red one is pushing back the partner and tries to stop the contact. Okay, so then we tested how the same rats are motivated to press the lever to get a food. And we found that inhibition of both projections affects the food response, that were less motivated than the control animals to press the lever and to get the food. They were interested in consumption of food, because when they get the pellets, when they got the pellets, they ate the food, but nevertheless motivation was lower here because they pressed the lever less frequently than control animals. Okay, but as I told you at the beginning, we were looking for some differences. We wanted to know that until now we saw that similar pathways or similar neuronal populations in the central amygdala are involved in control of social behavior and food motivation. But we wanted to find some difference, or well, to know whether there are some difference in the populations involved. And at this point, we decided that we need kind of a shortcut, because as I show you that there is like plenty of projections, so this system is very complex. We wanted to have some hints, and that's why we performed this experiment. We used the construct that is under control of Cephus promoter. So we marked the social cells and food cells, the same behavioral protocols as I described earlier, and then manipulated these cells when rats were pressing the lever for food. So the idea was that if the same here in the control conditions, when we activated the food cells, we expected that pressing the lever will be increased by optogenetic stimulation of these cells, because we assume that they are the same cells here and here. But here we wanted to see what happens when we activate these social cells, whether we also will see the increased lever pressing or maybe some different response. We assume that if they are the same, we should see similar effects like here. If not, we will see some other responses. And we did both the optogenetic stimulation and optogenetic inhibition. And what we saw was, firstly, I show you the inhibition. So here is the rat in which we inhibited the cells. So you can see that he's not very interested in pressing the lever. So the inhibition of both food cells and social cells resulted in this. It's not a freezing. It's just the rat was not exploring the environment and was not pressing the lever. But the most interesting part is in this activation, is after the activation of the cells. So when we activated the food cells, we observed what we expected, that is the rat was pressing the lever very eagerly, and it was like more pressing than in the control conditions. But when we activated the social cells, the rat started with pressing the lever, but then start to explore the environment instead of pressing the lever. So there was something different in the behavior of this and these rats. And of course, we quantified it. We did this experiment in a regular groups of animals with several animals. So what we saw, we confirmed that the activation of the cells, these food cells and social cells, have different effects on lever pressing. So we started to just a kind of a proof that there is some functional difference between these two subpopulations. So probably they partially overlap because the inhibition effects were similar, but there is also some difference. So as I've mentioned, we needed kind of a shortcut to know where to go from the central amygdala. When we manipulated the neuronal circuits in the central amygdala, we also manipulated outputs of these cells. So we decided to look at these potential outputs, and the shortcut means that we just extracted some parts of the brain after the training and stimulation and look at the level of metabolites of different neurotransmitters. And we look at we compare the social and food cell stimulation effects. And we found that we look at different parts of the cortex and the different subcortical structures. And actually we found the most profound differences between these groups in the anterior cingulate cortex and orbitofrontal cortex. This was interesting because, again, we had the changes in the cortices from which we started actually our experiments, not knowing that this difference exists. So we decided to look what happens when we manipulate these projections, and because we saw the difference in dopaminergic in the level of dopamine, we decided to look at the dopaminergic projections, and the central amygdala gives the dopaminergic input through the ventral tegmental area. So there is projection from the central amygdala to ventral tegmental area, and from the ventral tegmental area to anterior cingulate cortex and orbitofrontal cortex. But so we firstly decided to look what happens when we block this projection from the central amygdala to VTA, and we found that it profoundly affects the social contact. So the rats with blocked projection had decrease in both initiation and maintenance of social contact, and they actively blocked the attempts of their partners to initiate the contact. But there was no difference in the lever pressing for food. So it seems that this projection, manipulation of these projections affected only social interaction. Then we look at the type of the cells in the ventral tegmental area that received this input from the central amygdala. And in the ventral tegmental area, we have two main classes of cells. There are GABAergic neurons and dopaminergic cells. And we found that most of the projection ended up at the GABAergic neurons. As the central amygdala sends mainly inhibitory projections, probably what we have here is inhibition of inhibition, which means that when we inhibit these GABAergic cells here, at the same time we disinhibit the dopaminergic cells that project to the cortex. So then in the next step, we decided to look at these projections from the dopaminergic cells using TH3 rats, provided by Wojtek Solecki, thank you. And we look at the projections. Indeed, there are projections from the ventral tegmental area to both of parts of the cortex. We use the construct that allowed to manipulate this projection, but what was available was a construct that allows for activation of the projection. So here is activation, not inhibition, but activation, but it's specific for this dopaminergic projection. So what we found was increase in initiation and increase in maintenance of social interaction, and decrease of blocking. So it's like the opposite effect. And again, there were differences between the projection from the ventral tegmental area to the anterior cingulate cortex, which affected the initiation. And in the case of the ventral tegmental area to orbitofrontal cortex, it was increase in maintenance of social interaction. Both cases we had decreased blocking. So again, we affected the social interaction. What was interesting is when we tested the rats in the test in which they had to press the lever to get the food, there were no differences after this manipulation. So it seems that again, these pathways are more involved in social interaction than in motivation for food. So here you can see the motivated rat. So again, this is the red rat, which is the one in which we activated the ventral tegmental area anterior cingulate projection. And this is the behavior that is, the rat is very motivated to start the interaction, but his partner is not so happy whatsoever. So it's what is interesting here is that when we tested, actually this green rat, which was injected with the saline, so it's not, well, the control animal, actually didn't want to interact with his partner. So we probably we increased the motivation, but the way in which this red rat behaved was changed. And when we tested these green rats on the next day, drug-free, they were still afraid of their partners. So we probably changed the way how they interacted here. And this green rat had a bad memory of his partner behaving in the wrong way. So we can increase motivation, but it's like we at the same time, it's not the physiological way or normal way of how the rats interacted here. Okay, so to summarize, what we found here is that these sub-circuits in the central amygdala can be defined by their inputs and outputs. And we showed that this particular circuit that starts in anterior cingulate cortex and orbitofrontal cortex and involves central amygdala and ventral tegmental area, promotes social interaction. And this is different from what was shown before for the basolateral part of the amygdala, which mainly inhibits social interaction. So this is like positive driving force that starts social interaction. We have slightly different neuronal circuits for initiating and maintaining of social contact, and that the circuits that are involved in social drive and pursuing food rewards overlap only partially. So there is some overlap, but it's not complete. It is interesting because when we think about the problems with social interactions, like the ones we observed, for example, in autism, quite often the autistic patients have problems with social interaction, but not with food rewards. So you would like to manipulate some specific circuits to increase social drive, to social motivation, but not the ones that affect food, because we don't want to increase food motivation if it's normal. It's a normal physiological level. Okay, how much time do I have? Great. Okay, so here I would like to show you the story that we started recently in the lab, because as we discussed 2 days ago, of course, this very detailed mechanism, brain mechanism, are very important, and it's much easier to study them when we have simplified models, like behavioral models. The ones I showed you here are very simple. In the case of social interactions, we randomly take 2 animals and then put them together to the cage and expect that they will like each other. Yeah? But in the natural conditions, the animals, especially in the case of social interactions, they really care who and when they interact with. So there is some social hierarchy when we have a group of mice living in, or rats living in natural conditions. They form some social hierarchies. They interact with some animals, but not with others. They form some subgroups and so on. It's quite complex. So we wanted to see whether we can model such social group and social interactions in a larger groups of animals in more naturalistic conditions. So we started to use the system that we have developed in my lab some time ago, and the system consists of 4 cages that are connected with the corridors. The corridors are equipped with RFID antennas, and every animal has a transponder with a unique number, and it's recognized when it travels through the corridors. There is food and water available here in 2 cages, and we have like perforated partitions in the corners of other 2 cages, so we can expose the animals to some stimuli. Usually we use olfactory stimuli in the form of the bedding taken from a cage where other mice live, because mice are very olfactory creatures, so they are very sensitive to olfactory stimuli. So we can see how much they are interested in exploration of those stimuli. And the system is fully controlled, and we collect the data 24/7, so we can collect a lot of data. We can put a group of mice here. We usually have groups of 10 to 12 animals and observe them for days or even weeks. So this is still not the natural environment, but it was inspired by the field studies that show that mice love corridors, and they have nests and like chambers. They dig in the ground. They have kind of chambers connected with the corridors, and they really love such an environment. So they also like our EcoHub system. So, and we can follow their behavior, different kinds of behavior. Here I would like to focus on one of the measures, which I think is quite interesting. It's the following behavior in the tubes of this EcoHEP system. When one animal is closely following another, we call it following, and we measure how many times during some period of time. We usually, they have like dark light period 12 and 12 hours. So we usually, the dark period is active one for mice. They are nocturnal creatures. So we usually look at their behavior during this dark period. So we count how many times they follow other animals from the group in the tubes of the system during this 12-hour period. And what we found, well, we can show this behavior as the social structure or social networks using the tools that were originally developed for analyzing the networks in primates. Because primates, including us, humans, form such very similar networks. So what we show here is the number of followings, and the size of the circle show how well how often they follow each other, and the thickness of the line show how often in each part they follow each other. And what we see is that this behavior is quite stable. When we observe the animals for 10 days, what we see is, here are the data for just 4 days, but it's we did it for the longer time, so I know that it's pretty stable. So what we usually see here are the example of 2 cohorts of animals. So in the first day, the animals are very active, and it's still not very stable. They are in a novel environment. They don't know the environment, so they probably still stabilize their social structure. But during the following days, the structure is already stabilized, and it's the number of followings is pretty similar, and there is a clear individual difference. So there are animals that follow others much more than other animals from the group. And we also look at the correlation of this following behavior with the classical tests of social dominance, and we know that the ones that are the super followers, so the ones that follow other most, like the one here, or the one here, are the dominant animals. And the ones that follow other least are the submissive ones. So there is a correlation with the social hierarchy. But classically, social hierarchy is measured in so-called tube test, in which we put 2 animals into 2 ends of the tube, and we check which animal is pushing out another one. So it means that the one that is pushing out his partner is a winner, and we test each animal with each animal from the group, and with this way we have kind of linear hierarchy, because this test, the results of the test are linear. Here we see that the hierarchy is not so linear. There is there are some dominant animals, there are some submissive animals, but there is also a large group of animals that are in the middle. They are pretty stable, but not so far from each other. We also know that there are some differences in those structures. We put randomly 10, 12 mice, and we see that in some cases we have very clear dominant animals, but those super followers. But in some cases we have more egalitarian networks, so the mice are not so different from each other. So there is some variability here, and it's interesting because we tested here C57 mice. So the black mice that are commonly used in the laboratories, they are genetically identical. So it's we expected to have more similar results, but still we see these individual differences in this following behavior and in the propensity to be a leader or a dominant animal. So just one example, we did an experiment in which the mice were housed for some time in the EcoHEP. Then two of them were removed from the system, and one was offered 10% sucrose, which is very rewarding for mice, and another one was drinking just tap water. And then we collected the bedding from the cages of these mice after 24 hours of drinking either sweetened solution or tap water, and put the bedding here to the eco-hub where the other mice still were, and check whether they were interested in this social stimuli. And what we saw was that they were really interested, and they preferred to spend time with the bedding taken from a mouse that drank sweetened water. And so here, and it was interesting for them for a long time. We measured it for 12 hours, and for 12 hours those mice went back and back to this compartment, sniffing this bedding. And what we observed was also the increase of the following behavior. So the one that I showed you in the previous slide. Probably we don't know why it happened, but maybe they just tried to find where this reward was, because they could smell it here, but there was no reward in the system, so they were just exploring the environment, looking for the reward. This is just a hypothesis. We don't know it yet for sure. We are still exploring it. But what is interesting here, that when we look at this following behavior that increased after the reward was introduced, the increase was not random. It depended very much on the position of an animal and the social structure. The ones that were super followers before increased this following behavior most. So it's, I think that, this is the last slide. So I think that it's looking at the social structure in the case of social behavior and responses to social stimuli is important, because there is a clear difference in how the animals respond to such stimulation depending on their position in social structure. So as I said, this is the something that we started relatively recently, so we are still exploring it, and actually here we have more questions than answers, but I think that it is interesting. So here I would like to thank you for your attention and acknowledge input of several people from the Knapska Institute. The data I presented are the effect of a PhD project of a very gifted PhD student, Karolina Rojek-Sito, and I would like to thank our collaboration. Adam Hamet is with us, Wojtek Solecki is with us. Thank you very much. Thank you for your attention.
Thank you very much for this fascinating lecture. I think we have time for a couple of questions. So please raise your hand if you have a question, or if we have a question from the Zoom, we might also read them. Okay?
Thank you for this amazing lecture. It was really interesting. I have plenty of questions, but probably I will just ask one due to the time. So you showed that there those red and blue or green, I don't remember, rats, they followed each other. And when a red rat was following just the control rat, the control rat behaved in the way that it kind of repelled the red rat. Have you checked what happens if you put two red rats together?
No, we haven't checked it.
What would you expect?
I would expect some aggression. But we haven't checked it. It's an interesting question. We are exploring the problem further, so we'll see. It's a good idea for an experiment, but it's what we see is that they are very motivated to initiate the rats in which we activated this ventral tegmental area, anterior cingulate orbital frontal cortex projections. They are very motivated to be involved in or to start the social interaction, but the partners respond like, they are trying to avoid this contact, and for them it's I think it's aversive. We measured also ultrasonic vocalizations, and they vocalize at 22 kilohertz, which is a sign of a stress. We don't know which rat is vocalizing, but I think that it's quite probably that these control animals are just unhappy of what's going on. And so putting two such rats that are so motivated to start the social contact, but at the same time they behave strangely. They're like over-social. And I think that, with social interaction, there's like starting social interaction and being motivated to start it is one thing, but then you have to behave in a proper way. The timing is important. Responding to what partner is doing is important, and here I think this is change. It's very difficult to quantify it, but I saw like plenty of social interactions in rats, so I see that something is wrong, and I think here this is the case. But it's a good idea to put them together. We'll see.
So maybe a very small following question. Could it be that the treatment that you give to rats, so the optogenetic stimulation of certain circuits, change the plasticity of those circuits over time, and then those rats are actually different rats from the rats that they were?
I think this is something what we started to think about. Here in this experiment, every rat had this inhibition only once, because it's like we have two rats in each pair. So firstly, one was tested, like the projections were inhibited or stimulated during social interaction, and this food test we tested another animal just to avoid the double or repeated stimulation or inhibition of the circuits. But now we started to work with mice and with repeated chemogenetic inhibition or stimulation, and there are very pilot data still, so I'm not sure, but I think that what we see is that there are some differences when we stimulate the second or third time, so I think there is some plasticity here.
Thank you.
Okay, we'll have time for one question from Janek Rodriguez.
Actually, I have a similar question to the one before. So you would interpret the result from the central amygdala as a measure of social reward or something with social anxiety?
I think it depends very much on the circuit we are manipulating with, and this is a question whether, and we don't know it, we plan to look at it closer, because in the case of whether the projections to the cells in the central amygdala from the cortices are the same that send projections to the ventral tegmental area and to the cortices back. So it's probably there are different circuits, and of course, as I mentioned, there are not only circuits in the central amygdala. So it's like, I think that there are different circuits that are responsible for different aspects of this response. There's like motivation, but also some anxiety. Here it's like, at this moment, it's difficult to say, but I think that we can play or manipulate both the motivation and anxiety.
And we'll have last question from Zoom.
So it's a question from Zoya Haribina. Thanks for an interesting talk. Were there any sex-specific differences in your results?
Mm-hmm. So here we tested only males, which is a shame, but we had to start somewhere. And we know that social behavior is sex-dependent. So of course there could be some differences. We started to female animals, both rats and mice, are less tested than the males, and one of the problems is that behavior is more variable in females. And in such experiments I showed in the first part, when we manipulate the circuits, it's difficult to have larger groups because it's very time-consuming to perform such experiments. But in the ones in the EcoHub, the beauty of the system is that it's automated, so we can test more animals. And we started to look at the female mice and their interaction and their social structure. And I think that there is some structure. So we see stable structure also in females, but the social hierarchy is not so steep as in males. So I think it's an interesting question, and yeah, we started to look at it. But to go completely to like what we have in the natural environment, usually the sexes are mixed. So what could be interesting here is to put together some male and females to the same cage and see how they form the social structure in the conditions that are closer to what we have in the natural conditions. We plan such experiments, but it's like we need to do it in steps. So like at the moment we are testing both males and females, then we'll try to test both sexes.
Okay, excellent. Thank you very much.
Thank you.